most prominent organelles in the cell is the nucleus. The nucleus is very important because it acts as the control center of the whole cell. The nucleus is bounded by the nuclear membrane, which has tiny pores in the surface, so the nucleus can send messages to the rest of the cell. The job of the nucleus is to store the cell's genetic material. That genetic material can be seen in this picture as thread-like structures called chromatin. The chromatin is made up of DNA, which is like a blueprint used in construction. It contains all the instructions for building and running the cell. When the nucleus needs to get a message out to the rest of the cell, the DNA instructions are copied into a form called RNA. RNA can leave the nucleus through pores in the membrane and find a ribosome capable of reading the message. Every cell must have a copy of the genetic material in their nucleus. When a new cell is formed by cell division, very specific steps must be taken to ensure that the new cell has an exact copy of the DNA. Mitosis is the process by which cells divide. Cells divide in order to replace old or damaged cells. Cells divide to allow for growth. Cell division is what allows you to heal from an injury and how you've grown from an infant to the young adult you are today. The cell begins its life cycle in a state called interphase. At this time, the cell is going about its everyday business, performing metabolic reactions and growing. When it is ready to divide, it copies its DNA. This step is important so when the cell divides, each daughter cell will have one full copy of the DNA to store in its nucleus. After the DNA has been copied, the cell is ready to divide. Mitosis is the orderly sequence of events that take place during cell division to ensure that the two daughter cells are exact copies of the parent cell. Mitosis is divided up into 4 stages: prophase, metaphase, anaphase, and telophase. Let's look and see exactly what happens during the cell cycle. The cell spends most of its life cycle in interphase. At this point, the DNA is loose and unraveled in a form called chromatin. The cell grows and copies the DNA. Mitosis begins with prophase. During this stage, the loose chromatin condenses to form chromosomes. 2 identical chromosomes are attached by a centromere, and the pair are called sister chromatids. While the DNA condenses, the nuclear membrane starts to break down and disappear. The centrioles are activated during prophase, and spindle fibers begin to extend outward toward the chromosomes. The spindle fibers attach to the centromeres of the chromatid pairs, and the centrioles coordinate their movements as if they were puppets on a string. Cell division progresses into metaphase as the centrioles guide the sister chromatids to line up along the center of the cell. Anaphase begins as the spindle fibers pull the sister chromatids apart, and one copy of each chromosome heads towards the opposite ends of the cell. The cell itself begins to elongate. The final stage of mitosis is telophase. The daughter chromosomes reach the ends of the cell and begin to unravel back into chromatin. A nuclear membrane will form around each group of chromosomes to make 2 new nuclei. The center of the cell pinches inward. Mitosis is now complete, but cell division will not be complete until the cytoplasm has completely divided in 2. The process of dividing the cytoplasm is called cytokinesis. At the end of cytokinesis, we are left with 2 daughter cells that are identical to each other and to the original cell. Both of these new cells will immediately enter interphase and remain in this state until they too are ready to divide. Here is an overview of the cell cycle, beginning with interphase, cycling through prophase, metaphase, anaphase, telophase, and cytokinesis, and ending with 2 daughter cells. The fact that this is a continuous cycle for cells is why we refer to this process as the cell cycle.